more possibilities. We'll talk about hidden Markov models. That's a relatively long subject. You will see there are many possibilities. So it's in two parts. And so for this first part, we'll just look a little at, we start with example, because there are many possibilities with hidden Markov models. Often you don't think you could tackle the question with this type of model, but those are models which, let's say, give a good approximation in many cases. So I'll just show a few motivating examples. Quite a few come from the biology, because that's what I'm working on, but hidden Markov model we are using other stuff. And then we will again use a very famous toy example for hidden Markov models, which is a casino example, just to start to see how we can compute quantities with this example. And we will just start here for this course to see how we can compute the probability of a sequence which is generated by a hidden Markov model. Next week, we will see how we can do inference with this type of models and how we can learn parameters as well. So let's look at some motivating example. So I don't know if you already heard about this kind of data. This is data which is still used nowadays in clinic, which started to arrive before let's say high throughput sequences, and it's RAS-CGH data. So it's a technique where you can quantify along the chromosome of a patient, or let's say if you take a tumor sample from a tumor, you can see if you have a variation in the copy number with respect to a reference genome, to reference chromosome. And the idea behind that is that very often when you have tumor, when you have progression of a tumor, there are a lot of genomic rearrangements, and part of what makes the tumor active and being able to grow is that it changes its copy number. It allows it to deregulate its let's say its process. And so that's let's say that's the very first coarse-grained way of checking areas in the genome where suddenly you would have an increase in terms of DNA material. And that's you can see that here you have all the chromosome, and each point is like a region of a chromosome that is hybridized, and you do a ratio, you compare to background. So let's imagine you have a tumor sample from a patient, and then you have a somatic, you have normal cells, and then you compare, and you can see here in this region of chromosome 1, there is a log ratio which increase of log2 ratio of 0.5, which means 2 to the power. So it's like that you have it's not a double, it's so you have one copy more out of a diploid, and you can also see regions where it goes down. But so you can see you have a lot of points, and the question you would like to see is that, so where did it start to cut? Where did the cut start in the chromosome? Can I do that automatically and say, okay, here I have a cut, and can I estimate the number of copies? So what is exactly this log2 ratio? Is it like here 2.5? You see it's a lot. It's like 4, 5 more. Is it 1.5, etc. And so can we, from this sequence of points, so here you can see we can, the Markov part, the sequential data will be important. We have a sequence of points, and we want to estimate where there were change. So is there like where there were rupture, and if there were different values. Okay, so we want to segment the position according to those copy number. And we want to take, you see here from the modeling point of view, we want to take into account the fact that there is some variability associated to the point, and so that we can disentangle variability, like the measurement variability, from the real variability. Okay, so that's the first example, which can can be tackled with hidden Markov model. Another example from biological data. In fact, this is really one of the big success of hidden Markov model in biology for sequence analysis. And it really comes back from the first sequencing project, which started, the first genome was 95. It was influenza, the flu. and then human genome was in 2001. But for the human genome, we have 3 billion base pair, but only a very small fraction is coding for genes. In the first, as a first pass, so we were able to have a sequence, but we didn't know what will be transcribed and then translated in protein. And this was really like a first needle in a haystack problem. So in this case, you see, you just have a DNA sequence. You could look at just one strand. And the first question you could ask is like, can we find the genes? So here it's like on the bacterial genome, a gene will be a simple, what you call an open reading frame. So a sequence which is between a start codon and a stop codon, and which do not have any stop codon in the middle, and would have like the typical properties in terms of composition, which correspond to the usage of the codons. So I'm going fast. I'm assuming you have some basics in molecular biology, but the project will be on gene detection, so we'll see a little about it. And so then the goal is, you can see here, it's a fraction of a genome, and you would have a sequence, and you would have you would like to have a model which is predicting. You see here in green, this is the annotation. We know that there is a gene here from here to here. And we have in green, we have a probability of having the gene on the forward strand. So in this direction. And in blue, you can have a probabilities to have a gene on the other direction here on the back strand. You see that you can do this kind of prediction. It works here on bacterial genome. It can work well, not always, but that's the kind of prediction you can do with a hidden Markov model. So another motivation, which was in fact the first application of hidden Markov models. I can put the historical paper on the Moodle. This was for speech recognition, and this was used for a long time for that. And the goal is that you have an audio signal, so you have let's say wavelengths that are encoded. So you see frequency. So you want to do usually you want to go from those sound waves to different levels of abstraction to go back to words. So first, you try to get back the phonemes, so sounds that can be written in some alphabet, so collections of phonemes from the sound waves, and then from the phonemes you would like to re-infer word. And this is something which was well done with hidden Markov models before. I think now the best speech recognition tools that you have from the last 5 years, which really made a difference, are not based on this model anymore. Of course, and so I wanted to, and then a last example. I don't know if you use those smartwatches, but that's a very good example for summarizing what HMM can do. Is that you when you're checking your sleep pattern now, you can, all of those smartwatches, they have some accelerometer. I have one, in fact. So you have some accelerometer in the watch, so it can monitor the movement of your hand. Sometimes you can also have some idea on the heart rate, and it always provides you with some indication when did you fall asleep, and where were you in a very deep sleep, a light sleep, this REM phase of sleep, and where were you awake during the night. And a way to answer this question. So you see here, you have this, at every time point, you have a movement of the hand, and then you just want to classify that across time in those 4 states. That's a typical hidden Markov model problem. Okay, so we see like what was in common with all of those examples, and what's the typical use case of hidden Markov model is that we have events, YT, that we observe across time again. But now, instead of a Markov chain, which was saying that like our Markov chain was homogeneous, so we were observing the values always under the same distribution. Now we want to keep this sequential aspect, but we want to classify. We want to think that they are like the observation we are getting their value depends on some hidden categories that we don't see. So we want to annotate, we want to classify into some categories given some observation. But so the difference with the other classification is that now we observe values like along a sequence, so across time. So there is some kind of time dependency between the observation. And of course, that's easy. So we know if you're in deep sleep, we'll move less. So the observation will change. They depend on the category you're thinking of. So like the movement you're doing. And so how can we? What will be a way to tackle this problem? To look at this problem. So the easiest, or like the most simplest model one can do with sequential data is to say, okay, we will have latent variables. We will have those category variables that will determine how I observe the values. Okay. And I will put a constraint on my latent variables. I will not say that they are independent. I will say that they are following a Markov chain. And so that's simply the trick. So you see that's really easy, but you see also that those graph of dependency that we used to draw, now it's getting a little more structured, but that's still a very simple structure. So that's the category, the hidden state changes according to the Markov chain. And at each time point, given the hidden state, we generate value. Okay, so we write an explicit conditioning. The state determines the distribution for the value. And so for the states, you see, like from the example we had, the state that can be which stage of the sleep you are. So those 4 stages of sleep. And given the stage of sleep, of course, the movement value you will get, so those will be real valued, they will different. Again, so in the case of RACGH, the hidden state will be the copy number. Okay, and for the case of the genes, now we have a process which will tell us I can be, so if I'm on a stop codon, then I will do only those 3 stop codons which are possible. That's the only types of letter I can observe. If I'm in another part of a gene, then maybe I just have random values. Okay, and so those are the observation that we get. And the hidden Markov model, you see, it's a way to structure your sequential data so that now we put some structure here in the hidden part, and we will specify how it goes from the hidden to the observed, and with the model we will be able to learn the parameter and go back from the observation to the hidden value that gave rise to this information. So to do annotation with that, okay? And and they are like so we will see that again later on with the example. But so we can do many things in fact with those Markov chain. We can annotate, but we can also, like in the RACGH example, you know, the goal is to find stretches, groups of position with a given like intensity ratio copy number. So the first control we can do is that by staying in the same groups of states, we can smooth our signal. So we have a noisy observation, but we can smooth, say, okay, all those observations, they vary a lot, but in fact they correspond to some stages. And we can put, as we just saw, we can put some knowledge about how much time we expect that them to stay. Okay, so that's the first thing that we can do with a Markov chain. We can smooth, but we can also, because of the automaton structure of a Markov chain, every time you take out links between states in your Markov chain when you have a lot of states, all the links you take out, they create a lot of constraints, but they can create combinatorial constraints. This is something if you have an automaton, But I think that becomes quite clear. But that's also something that's completely encoded in the Markov chain, that we can integrate those hard constraints quite explicitly. And so, for instance, for the gene example, we know when we're in bacterial genome, the only way to end a gene is to be on a stop codon. So the gene should only be detected, going back from a stop codon. We go back and we see if we have what we call an open reading frame. So a sequence without stop codon in the same frame up to a start codon. And so those are really strong constraints, but we will see in the project with Hidden Markov Model. But those constraints, they can be integrated quite explicitly in the model. Okay, so let's have a look at a toy example now. This was a motivating example. Now we will go back to a very simple example, which is made up to be a real Markov chain, just to see how it works. So the most simple example is that you will have only two types of. So we observe again a very simple. We observe a dice result. Okay, but we will make the hypothesis that we have two types of dice. In this case, you have a traditional dice which gives the same probability to each of a number from 1 to 6, and you have a loaded dice, and this loaded dice has a 50% chance to get a 6. Okay, and so of course, if someone starts to use the load dice in like for a few times in a row, you should see a lot of 6 occurring. Okay, and so that's the idea behind the model. We start with a fair dice, and we will randomly change the dice at each turn, and we change the dice according to a Markov chain. So that's the goal. We're constructing a hidden Markov model. We'll keep the loaded dice with 90% chance, and so we can change from the loaded dice to the fair dice with 10% chance, and we keep the normalized with probability 95%. Okay, so what are the parameters somehow if we want to represent that simply? We have a starting distribution we need to know. So we start with a fair dice. So probability of the first hidden state equal fair dice f is 1. And then when we have that, so we have the change, the Markov chain telling us how we move from the fair to the loaded dice. So those are all those arrows. And then now, conditionally on this Markov chain, we observe the values of the dice. And you see here we have 1.16 probability to have each of the letters. And with the other one, we have 10% probability for a number from 1 to 5, and 50% for number 6. Okay, so I let the summary of the model here. So let's just see how we would generate a sequence. Sorry, I did not manage to do an animation for this one, but let's. You all know how you generate sequences. You both know how you generate sequences when you have on the computer, when you have a random generator. So we start here with the first dice. We know it's a fair dice. Okay, so then we draw a random number between 0 and 1, and depending on the value, if it's below 1 out of 6, so 0.16, we will get 1. If it's between 1 out of 6 and 2 out of 6, we get 2. Between 2 out of 6 and 3 out of 6, we get 3, and etc. So we have a first fair dice. We draw this value. It's between 1 out of 6 and 2 out of 6. So we draw 2. Then we have to decide, do we keep the fair dice or do we move to the loaded dice in the second step? We draw again a random number. Here it's 0.26. This is below 0.95. So I keep the fair dice, and I generate, I draw again a random value to generate the value of the dice. 0.51 is between 3 out of 6 and 4 out of 6. We get a 4. We draw again 0.005. This is below 0.95. So we move to the fair dice. 0.54, it's the same. We draw a 4. Here I draw 0.98. This is above 0.95, so I move from the fair dice to the loaded dice. Okay, and now I have a loaded dice, so I'm using this table of probability to generate a value. I get 0.65. This is more than 5 out of 10, so I draw a 6. Okay, etc., etc. And we can generate the whole sequence like that. Okay. And now we can ask yourself, we see that's very simple. In fact, what we do, we go down the conditioning to generate the sequence. Now we can ask ourselves in the same way, how can I compute the probability of such a sequence, given that here I know what are the hidden states? And I can just compute here the probability of having a 2, given I have a fair dice. So that's the first one. I know I start with a fair dice, so I have probability of getting my 2, given the fair dice. Then I multiply that by the probability of moving from fair dice to fair dice. Now I generate a 4, given I have a fair dice. I compute this probability. I do the same for the next one. I move from fair dice to fair dice, and I generate my 4. And etc. Then I move from fair dice to loaded dice, this probability, and etc., etc. I generate a 6 given that I have a loaded dice, and then again I compute the probability of moving from loading dice to loaded dice times the probability of having a 6 given I have a loaded dice. Okay, so you see here the computation of the probability. We could write that in a formal way, but that's something we will do maybe for the exercises. The computation of the probability of a sequence when we know what are the hidden states, what are the types of the dice. This factorizes, because of the structure of the dependency we put, this factorizes in a way which is similar to the Markov models, except that we do that in 2 steps. We move in the hidden process, and every time we fix what we see in the hidden process, we compute the probability of generating the data. Okay, and in fact, that's something I will add as an exercise, and we will say a little later on. If I know what are the dice, I can construct a Markov chain, which would have many states, which will be the Markov chain on the value of the dice and the identity of the dice. This will be a big Markov chain, and you see that our hidden Markov model, if the state is not hidden, it's equivalent to it can be cast into a Markov chain. Okay, but so that's not the problem in general. The main point of having those models is that we don't know what are the dice which generated the sequence. So the first question we can ask now, I observe this sequence. I know that I have this hidden Markov process behind. We generated the sequence. Can we first compute the probability of a sequence? Independent, no matter what are the hidden states we have behind. So we want to marginalize, we want to sum over all possible hidden state allocation. So how can we compute this probability? This is what we will do today. That's very practical, but that's not directly answering the question. The question that usually you want to do when you're doing hidden Markov model, you want to annotate the sequence. You want to know, for instance, here, when was the loaded dice used here in this sequence of dice values? And this is something we'll see next week. Just to summarize now the example on the casino, what are the parameters we are using, and the terms, the word we can use, how we can describe what we are looking at. We have an observed sequence. It's usually I will use y as a variable to name that yt, and we have a hidden state sequence. I use s here, but sometimes it's x. Okay. For so that's what's on the observation. For the model, we have what we call the emission probability, because we emit values, we emit observation given the hidden state. And so we need emission probability for each of the hidden states. And then we have a transition probability between the states. Okay, and so the basic question of the hidden Markov model is just what I just wrote. How can we annotate a sequence? So how do you write that in a formal way? I mean, we already saw how it works, but it's just to fix the idea. We say that the process on ST, the process which is hidden, is a Markov chain with the transition matrix. I mean, I wrote it alpha, but we still use A. So we have a Markov model on the hidden states, and the observation YT to observe a sequence at times t only depend on the hidden state st. So we don't need to know what happened before in terms of hidden process to know what's happening for yt. Sometimes, if you want to, you can take a little of context from the observation into account. And you speak of order 1 hidden Markov model, but I think we will not get too much into that, because it's just making the formula bigger, but that doesn't change much about the understanding. So the main point here, if we focus on we look on the order 0, is that when we observe our observation, I just need to know what is my hidden states to describe how my observation is observed. And we'll see soon that in terms of computing probabilities or likelihood, this will allow us again to do a lot of factorization. So if I go back, I should have put it here, sorry. It's this structure of a graph that we put, which tells us exactly that here, that yt just depends on st. This structure will make a huge difference. It will be what makes possible to factorize the computation for the likelihood. And okay, and so what are the big questions that we will want to answer in the next course is the evaluation. What is the probability of a sequence? If you know the hidden state, we just saw that it's relatively easy. It's an extension of we can do that by conditioning, a little like for the Markov models. We will see now how we can compute the likelihood of a non-annotated sequence. The next question is what you call decoding. So from the sequence, what is the most probable allocation of hidden states? So either the most probable path in the Markov chain or the most probable hidden state for each position. And there is an associated question, which is how can I learn parameters from a hidden Markov models? And we'll see that what's really nice is that if your hidden Markov models has enough structure, you can learn with EM again. We can learn parameters of a model without having annotated examples. So we can do both at the same time. We can learn the parameters and annotate the sequence. And those are questions that here I cast into the framework of hidden Markov models, but all models, also the models we will look at afterwards, where you have hidden variable you want to complement on, they will work on answering this type of question.